Good morning. Hello. Welcome to the mini course of the role of mineral surfaces in prebiotic chemistry. Welcome, Dr. Lambert. Before we start to event, it's important we have to know in case of earthquake or fire or any incident, the emergency exits are located on your left. If necessary, please evacuate most calmly. Thank you. Welcome to Minicurso, which will be given by Professor Jean-Francois Lambert, whom we thank for sharing his knowledge and for accepting our invitation. Ahora, presentaré a los doctores en la mesa, que también son anfitriones de este evento. Primero está el doctor Alejandro Heredia, que es el coordinador del Departamento de Química de aquí del Instituto. Gracias, doctor. Nos acompaña también el doctor Arturo Becerra, del posgrado en Ciencias Biológicas, y nuestro director, el doctor Alfred Barry Uren, quien les van a dar unas palabras de bienvenida a nuestro curso. Muchas gracias. Adelante, doctor.
Good morning, everyone. It is a great honor for our institute to extend a warm welcome to the distinguished Professor Jean-Francois Lambert from Sorbonne University in France, who is joining us today to teach the mini course entitled The Role of Mineral Surfaces in Prebiotic Chemistry. Professor Lambert is an internationally renowned researcher in the study of minerals and their diverse applications across fields such as surface chemistry, hybrid materials, and notably prebiotic chemistry. He currently carries out his scientific work at the Laboratory of Molecular and Structural Archaeology at Sorbonne University in Paris, one of the world's most prestigious academic institutions. Professor Lambert's research lies at the fascinating intersection of chemistry, geology, and biology, with a profound focus on one of science's most fundamental questions: how did life arise on Earth? His work seeks to shed light on the processes by which simple inorganic molecules could have transformed into key components of biological systems, highlighting the essential role that mineral surfaces may have played in the transition towards life. This line of research traces back to the pioneering studies in prebiotic chemistry, which began in the 1950s with the famous Miller experiment, and today continues with a level of sophistication and depth that brings us ever closer to understanding this great mystery. Beyond his impressive scientific contributions, Professor Lambert has had an exemplary academic career. He obtained his PhD at the Catholic University of Louvain in 1987, held postdoctoral positions at the University of Wisconsin in Milwaukee and at the Soil Science Laboratory in Versailles, and since 1990 has been a member of the Surface Reactivity Laboratory at UPMC. In 2004, after receiving his professorial chair, he co-founded the research group on interfaces in biological environments, alongside Professor Claire-Marie Pradier. His presence here also coincides with his participation in the second Franco-Mexican School on the Origin of Life, which will take place at the Colegio Nacional from April 23rd through 25th, a venue for high-level international scientific collaboration. For us, it is a privilege to welcome him to UNAM. His visit strengthens the academic ties between Mexico and France and inspires our community to continue exploring the frontiers of scientific knowledge. Professor Lambert, we warmly welcome you. We are truly grateful for your willingness to share your expertise, your passion for science, and your commitment to training the next generation of researchers.
Thank you very much, Professor, for this kind introduction. And I would also like to thank Professor Alicia Negron and Antonio Lescano and other members of the faculty for allowing me to be here today to, as it has been said, reinforce the ties between Sorbonne and UNAM, and also to take part in the coming French-Mexican School on the Origins of Life. So, I have seen this nice poster that was devised by, I suppose, the communication department. It's full of sound and fury. It's a representation of maybe the state of the Earth 4 billion years ago. I'm afraid that my presentation will be a lot tamer than that, actually, and you might be a little disappointed. So, just to tell you what I'm going to speak about, this is going to be a talk about the scientific approach to the origins of life, a little bit, and especially the place of chemistry. I see the place of chemistry in this kind of research, and mostly the place of surface chemistry, which is my original domain of specialization. There will be 3 different parts: a short introduction on the origins of life research, and then I will spend quite a lot of time telling you about surface science, because I was told that the audience would be constituted mostly of undergraduate students, and that maybe some background on this aspect of things was necessary. And in the third part of my talk, I will speak about the applications, logically, of surface science to the study of the origins of life. Let us first look at the stage. On the left-hand side, you have the Earth just after it was, not to say created, but appeared and started orbiting the Sun, maybe 4,400,000,000 years ago, where there was an ocean of magma. And then on the right-hand side, you have the situation a few 100,000,000 years ago, which is not so long after all. And it has changed a lot. You have an atmosphere, although it contains no oxygen. You have some oceans. You have some islands, not yet tectonic plates, but definitely you have some islands above sea level. And the artist has represented also an old form of life, an ancient form of life, stromatolites. So within a few 100,000,000 years, life was here, and it was here to stay. Okay, so how did life begin? And first, isn't it a question for biologists and not for chemists? Well, actually, we do have in biology a very potent theory of the evolution of life, and it started as Darwin's theory of evolution, which stated that the evolution was based on random variations, mutations that were inherited and then selected according to whether they were harmful or advantageous for the survival of the fittest. It was only much later that the molecular basis for this theory was understood, and that starting in the 1940s, the neo-Darwinian synthesis emerged, in which variation was interpreted as random mutations of bases in DNA. And after that, DNA was translated in proteins, and it's those proteins that are actually selected for their value for survival in a cruel world. Okay? So for this theory to work, it supposes that the basic machinery of the cell is already in place. And this basic machinery is already highly complicated. Here you have the well-known DNA double helix with with the double strand of complementary bases. This is a detail on 2 pairs of such bases, and it's a very long and very complicated molecule already. You also need to have a machinery to duplicate DNA, that's for the inheritance, and that involves a lot of proteins interacting with the DNA strands. You can find some details in the basic biology courses. And then you need another machinery to translate the message in the succession of bases into a protein, and that happens in the ribosome. And unraveling the functioning of the ribosome was a very long affair, and it was worth at least 1 Nobel Prize, and it was very much worth it because once again, this is an extremely complicated molecular machine. In short, all of these elements of cellular machinery are not things that you expect to find spontaneously formed in a test tube. More generally, if you look at the cell, which is the basic unit of life nowadays, and if you could see the individual molecules, you would realize how complex that is. You have here the outer membrane, you have a flagell, which is a machine, another molecular machine that allows cell motion. You have a lot of proteins in a highly concentrated water solution, and you also have some strands of nucleic acids, and then you have a lot of other structures. You see that this is already extremely complicated. So, of course, you would expect cells to have come together at some points and to have been derived from something that was much simpler. So what was there before the cell? That's the big question. Now, there is another aspect of evolution theory that has bearing on the increase of complexity that must have happened sometimes. You see, it's a basic principle of the theory of evolution, is that it has no inbuilt purpose. Variations, changes, mutations can just as well increase complexity as decrease it. Only there is a so-called wall of complexity. We have seen that cells were very complicated structures. If the complexity of these structures is decreased by much, then it doesn't function anymore. It's not able to reproduce anymore. So that explains why complexity increased overall from the first cells that you see here. That's in this graph, you have a complexity scale on the x-axis. That was the situation some Precambrian, in the Precambrian era, which was maybe 1 billion years ago. You only had the simple cells like bacteria. Now you have some things that are objectively more complicated, including the dinosaurs, including ourselves, and I don't know if we should be at the most complex end. Certainly we would like to think that. And that can be explained because, you know, some random mutations increase the complexity, but those that decrease the complexity simply died out because the cell became too simple. That's also a conclusion of another direction of research, the quest for minimal cells, which is carried out by some scientists like Luigi Luisi. It's a top-down approach in which you take a cell and then remove things until it doesn't work anymore. Okay, so there is really a wall of complexity. And then we have a problem because evolution doesn't explain the existence of the precursors of the cell. So then, couldn't we use the same approach as the paleontologists, for example? Or couldn't we use some devices of molecular biochemistry in order to understand how this complexity came into being? Well, One first direction, it's also top-down, start from what exists now to go back to states of lesser complexity. That would be the analysis of either the genome, that is the set of bases in the DNA, or the proteome, the set of amino acids in a protein. And indeed, some biologists can do that, have done that, and they have put into evidence the existence of some primordial biopolymers, like in the crucial site of an enzyme, for example, there may be a highly conserved set of amino acids, or there might be some amino acids that can be changed into one another by a simple mutation. And you can see, for example, that very simple sequences like these, D means aspartic acid, it's one of the amino acids in the protein chain, X is an undefined amino acid, and G is glycine, so that's only 2 amino acids, but they are very often found in the important sites of proteins. And that gave rise to a hypothesis that is called the GADV hypothesis, divided by Ikehara, saying that, okay, based on these highly conserved domains, it's conceivable that proteins started in a simpler state with, instead of 20 amino acids, a limited set, limited to only 4 of them, glycine, which was found here, alanine, which is just a little more complex than glycine, aspartic acid, which is D, and valine. Okay, there are some variants on this hypothesis, but it's indeed a promising direction for research. However, that still doesn't tell us how the machines that build the proteins came into being. So couldn't we just try to find some fossils, right? If you ask the people in the street what they think about the evolution of life and how to find out about it, they would think about something like that. Fossils of a T-Rex, and that's how we know how they were once Tyrannosaurus on our Earth, because we have found their remains. They have been modified, okay, but they can be easily recognized as some living structures. So could we find some fossils of things that were there before the first cell? Well, it doesn't sound like an easy task for several reasons. First, they certainly were very small. These structures were smaller than a cell. They were smaller than the size of 1 micrometer. I'm sorry, there is a misprint here. It should be a mu. Second, they should be less complex than a cell. Maybe if you see some cells of eukaryotes now in fossil materials, you can recognize the nucleus and maybe the mitochondria, but these do not even exist in bacteria, and much less in precellular structures. They would have actually very little structures, just a little blob. How can you say that it was something alive? Furthermore, that came before the first attested fossils of early life, so that means that they must be more than 3 billion years old. Okay, and they had a lot of opportunities of being modified. And yet, some people have claimed to see very old structures, like this is from the work around the year 2000 by a Schopf, and he claimed to have found, you see, these structures in Australia, in the Pilbara Craton. That's a photograph of these small structures. That's a drawing in which he evidences what he thinks are very small cells, and he says it looks like a filament of cyanobacteria nowadays. But this work has been heavily contested, and a little later, Martin Brazier claimed that actually these structures are not the remains of living beings at all. they just look like it, but they can be mimicked by purely chemical reactions. And I think there were very heated exchanges in some congresses between the proponents of the two different views. So you see, even for the first cells, you cannot be sure if they are really cells or not. There are still some elements of fossil records that can tell you, yes, life was there. There are some molecules that have very typical structures not found outside living beings, like the hopanoids. They keep the structure of these polycyclic molecules that you can find in some hormones, for example, in modern life. And they are very stable towards degradation. So you can find them in very old samples and say, okay, I have some evidence of life being here today. And that's certainly useful to tell you when it started. But that will not tell you how it came about to be, how it all got together. The same can be said for isotopic measurements, isotopic excesses, the ratio, for example, of carbon-13 to carbon-12, can also tell you if this matter was living or not. But that's only one small piece of information, and it gives you no hint of the complexification of matter. So more or less, when we want to understand now how the first steps towards life happened, instead of a top-down, we should adopt a bottom-up approach. We should start from the bottom instead of from the top. And I will use this picture quite a few times in my talk. You can read it in two ways. You can read it in an analytical way, that was René Descartes' approach. You start from something very complicated like a human being, and you decompose it into its components. And you see that there are some organs, some tissues, and then there are some cells, and it was understood in the 19th century that the cell was indeed the basic unit of everything that's living. But that's not all. If you go inside a cell, you'll see some functional systems. As I said, in eukaryotic cells, they are often kept in specialized organoids like mitochondria for respiratory systems, or the nucleus for the genetic material. But even in a bacteria, you can see the chromosomes. And if you analyze these systems chemically, you see that they are made of specific macromolecules, biomacromolecules, like the DNA that we have already seen, and the proteins that I have also mentioned. And I could also speak about the complicated sugars. These are very typical biomacromolecules, and themselves they are made of the catenation, the connecting in chain of small monomers. In the case of proteins, these are the amino acids. In the case of DNA and RNA, they are the nucleotides. Okay? And these molecules, as you know, are made of atoms, themselves made of subatomic principles. So if we analyze life in a descending order, we get from the large-scale organism to the smallest components. And it's quite logical to believe that the appearance of life happened in the opposite direction, starting with the simplest structures and going all the way up the scale, and that you had this first step, and then this one, and then this one. Okay, that's indeed a rather fruitful and logical approach. So let's try to do it. The first step, explaining how atoms were made, and that's very necessary to have living being because we are made mostly of carbon. Carbon is obviously necessary to form the backbones of our molecules. So how is carbon formed? That's not a question for chemists. It's an interesting question, certainly, but it's a question for physicists, and they understand it quite well. it would be a very good subject for a talk to speak about stellar chemistry, but that's not what I'm going to talk about today. The next step, going from atoms to molecules, well, that is chemistry. Chemistry is the science that explains the molecular level of organization in our world. After that, if you go from small molecules to macromolecules, and from macromolecules to functional systems, as far as I'm concerned, that's still chemistry. These are some questions that can be tackled by the concepts and by the methods of basic chemistry. So basically, this is what I'm going to be interested in today. These transitions from the atoms to the small molecules, the macromolecules, and the functional systems. Now, I'm not going to make a very thorough introduction to the field of the origins of life, if only because there are some people here at UNAM who know at least as much as me about this subject, and who certainly give you some lectures and courses about it. But I could just mention the old controversy. What started first? And if you want to know how life appeared, you should know how life is defined. And that's not an easy question. But most people would agree that two of the defining features are the information that is held in the genome and the metabolism, the ability to self-construct. Sometimes it's called autopoiesis. So which one came first? That's a question that has been around since the first scientific thoughts about the origins of life. And in particular, there was early on a controversy between these two great scientists, Oparin on the one hand, who was a Soviet scientist, and Haldane, who was a British biologist. And to schematize a little bit, I know it can be viewed as a caricature, but the idea of Oparin was that life emerged from a set of self-sustaining chemical reactions. In other words, a proto-metabolism. And in fact, it's hard to say how much it was his own idea and how much it was determined by the fact that he lived in the Soviet Union, a Marxist state, in which it was believed that in the terms of dialectics, infrastructure determines superstructure. So if he had been in opposition with this idea, he would probably have got into trouble, I think. On the other hand, Haldane was of the opinion that life started with information of molecules, molecules that became able to store information. And certainly it would seem that the development of molecular genetics later has put a lot of weight behind Haldane's opinion. But recently, there have been some new developments that seem to give, again, some validity to the idea of metabolism first. I will not insist on that, and for one, Antonio Lascano would be able to speak a lot better than me about these controversies in the history of science, in the history of reflection on the origins of life. But basically, this opposition between metabolism first and information first can be summarized by the opposition between two sets of macromolecules. Proteins are the efficient molecules in the cell. They are the molecules that do things, that synthesize materials and so on, that are responsible for the existence of solid structures. While, as you know, DNA stores the information. And knowing which one between proteins and DNA came first is very much a chicken and egg question. Because in the cell today, if you want to make some proteins, you have to translate the message of DNA. So DNA has to be there first, right? But if you want to duplicate DNA, you need some proteins. So proteins should be there first. How can you come out of this dichotomy? Well, a solution was proposed already a long time ago, already some 40 years ago, and that was the very successful idea of the RNA world. RNA, ribonucleic acid, is not exactly the same as DNA. There are some small chemical differences between the two macromolecules, but it's a molecule that can do everything. It can store information. It has a sequence of nuclear bases, just like DNA, and it can also act as a catalyst, just like enzymes do, but enzymes are proteins, and RNA is not. Okay? That idea was popularized, and I'm sorry, but the name of the author doesn't seem to appear anymore here, but it was popularized in 1986, shortly after the first ribozymes, the first RNA-based enzymes were evidenced. And it's one possible way to explain how two sets of interdependent macromolecules came to exist. The idea is that at some time, the two functions were carried out by the same type of biomolecules. So then, where does the RNA come from, even assuming that we accept this RNA world hypothesis and that a single molecule was at the origins of life? Well, you still need some chemistry to assemble RNA. You see that it's also a quite complicated molecule. Here you have the strand that is made of ribose units connected by phosphate groups, and here you have the rungs of the ladder, so to speak, that are nuclear bases. So it's a polymer of nucleotides, just like DNA, and we can expect that these efficient ribozymes were formed from simpler chains that didn't do quite a lot, but had already some structural complexity, and that themselves, they were derived from nucleotides. Here you have the ATP molecule. By polymerizing ATP, you would get a simple RNA. By polymerizing with the other nucleotides, there are 4 of them, then you could get a molecule as complicated as that. And this is, as I said, ATP, but structurally it's also not very simple. One ribose unit here, one nucleobase, one phosphate group. How did it all come together? That is already something more complicated than the typical molecules that you get, for example, from astrochemistry. So that's what prebiotic chemistry deals with. Propose some reasonable scenarios to go from simple molecules towards nucleotides and towards later polynucleotides. Now, of course, each one of the steps that we invoke must be compatible with the basic laws of chemistry. Okay? It's not because we are moving on to something more complex that the laws of the underlying levels of complexity are not obeyed. If you are studying chemistry, the laws of chemistry must be compatible with those of nuclear physics. And if you are studying biology, the laws of biology must be compatible with those of chemistry. So, in some cases, as we will see, it may need chemistry in special environments. And one of these environments would be the interface with minerals. So this is another cartoon representing the primordial Earth, where you have the ocean and its islands. It's already warm enough, well, it cooled down enough so that there would be liquid oceans. And here you have some meteorites or maybe comets falling down. Well, it seems to be a heavy shower, but of course it's an idealized vision. And maybe they bring about some simple organic molecules. So now what you have is organic molecules in a water solution, in an aqueous solution, in contact with some minerals. And so what you have is a solid-liquid interface. You have a mineral surface, and that's why mineral surfaces could have played a role in the origins of life. Now, what minerals? Well, there is a long list of them. This has been studied by geochemists such as Bob Hazen, and he proposed a set of several hundreds different mineral phases with different compositions, different chemistry. So that's a very large set of minerals that we can play with. And what I'm going to do next is to tell you about the complexity of mineral surfaces. Okay. This I will do in 4 parts. First, asking who does surface science. That's not only or not primarily people interested in origins of life. Then I will try to give you a very general idea of what surfaces look like. And then I will speak about adsorption, which is the reaction of molecules coming from the solution and the mineral surface. And finally, I will speak about heterogeneous catalysis, the catalytic effect of surfaces. So first, who does surface science? Well, I should say that there are 2 main stems in surface science research. There are the people researching planar surfaces, which are very simple from the, I would say, structural point of view. You have just a plane that is cut into a bulk material. And that's a field that has been developed a lot by physicists interested in material science who wanted to make, for example, semiconductor structures, right? That was a big impetus behind the development of complicated laboratory structures that are now found in surface physics departments. And then there is another direction of research which is much older. It didn't need these complicated structures, just some basic chemical abilities like measuring the amount of a substance in solution before and after contact with a mineral surface. Well, that was studied by colloid chemists, by geochemists, and many others. And that's the basis for a thriving industry. What you see here is a reactor used in petroleum chemistry, very large-scale reactor, and it uses heterogeneous catalysis. And heterogeneous catalysis is based on surface chemistry. In order to be a little more precise, divided surfaces like powders are studied first by colloid chemists who study dispersions of nanoparticles of minerals in aqueous solutions. And as I said, that's mostly a physical chemistry and a macroscopic approach. You only need to be able to make a chemical measurements in the large scales. We can say that this field has existed since the 1920s, if not before. For example, in soil science or in industrial chemistry, so that was very much motivated by the practical applications. And the geochemists, of course, are using the same kind of concepts to understand the evolution of our world, to for example, understand weathering, the modification of minerals by rain and other similar processes, to understand the chemical cycles of the elements, the transformations of minerals, and so on. We can say that these people are studying wet interfaces because most of the time they are interested in surfaces in contact with water. And then in the field of heterogeneous catalysis, well, we are speaking here mostly of dry interfaces. Because heterogeneous catalysts are used most often in the presence of gases, because they need high temperatures. It's also based on a macroscopic approach with a lot of weight being put on the understanding of chemical kinetics, of course. If you do catalysis, it's because you want reactions to happen faster, so you want to understand the kinetics. And it was driven, as I said, by the necessity of having some efficient industrial processes like oil refining. On the other hand, I said that there are two communities of scientists who are interested in surface science, the other one being that of physicists who are interested in flat surfaces. It started actually with electrochemistry. If you have an electrode, it's mostly a flat surface. And in order to understand the electrochemistry, well, you had to think about the surface science of these flat electrodes. But later on, and I would say it started in the 1950s and especially in the 1960s, some physicists started to approach flat surfaces from another point of view. They tried to understand really at the molecular level what was happening, and they developed a lot of new techniques. I will not have time to explain all of them here together. Some of them are familiar to you, certainly if you are working scientists, but also for students, like X-ray photoelectron spectroscopy. And some others are more specialized, like LEAD, which is low energy electron diffraction. IRAS is a variety of vibrational spectroscopy, of IR spectroscopy, that can be applied to surfaces, to reflecting surfaces. STM, of which I will speak a little more later, is scanning tunneling microscopy, and AFM is atomic force microscopy. Both of them are examples of near-field microscopies, and they allow an atomic level approach of the surface. And after a while, it really became a powerful characterization tool, and it started to allow understanding the intimate working of heterogeneous catalysts, for example. I can cite here two names. Of course, Günther Ertl, who got a Nobel Prize in 2005, if I'm not mistaken, for his work on flat surfaces, and also Gabor Chaumuriai, who might also have obtained a prize according to some people knowledgeable about the field. Now, all of these techniques, especially LEAD and STM, are more comfortable with the single crystal surfaces, and they were originally developed for them. But now there are also some applications to flat surfaces which are not crystalline, which are not regular, not periodic. And that involves, for example, biosensors, or that involves other biomedical applications. Some of the techniques I've mentioned can also be applied to non-crystalline surfaces, chiefly XPS and IRS and AFM. Okay? So now, after this maybe sociological introduction, what kind of scientists work on surfaces? Then we can ask, what do surfaces look like? And I will start with what's easiest to represent, the surfaces of crystalline solids. Now, of course, if you are going to cut a surface in a crystal, what the surface looks like will depend on the arrangement of atoms in the underlying three-dimensional structure of the crystal. Let's take a simple example. If we want to cut a surface into platinum, well, platinum, like many transition metals, has this simple face-centered cubic structure. This is the unit cell, and these are the positions of the platinum atoms on the vertices of the cube and the centers of each face. Now, what the surface will look like will first depend on how you cut it, how the cutting plane will be oriented with respect to the unit cell. And for this, in crystallography, we have some tools. We have the Miller indices, which allow you to define the position of a given plane with respect to the three basis vector A, B, and C. For example, plane 1, 0, 0 will cut vector A at A over 1. So it will go through this atom, and 0 and 0 means that it's parallel to B and C. So plane 1, 0, 0 is this plane. So now it's very easy to see what the 1, 0, 0 surface will look like. Okay. Here, I have just taken this face of the cube, and because the crystal is infinite, of course, it would go on periodically in the same way in the two directions of space. So you see that you will have a square surface. It can be defined here. Oh, sorry. Yeah. You have a square surface. It can be defined here by the intercept of the cutting plane with the three-dimensional structure, but it can also be defined by a smaller square. If you just repeat this small black square here, you would get the small surface. Anyway, you have a square symmetry. Here I've just rotated the previous view by 45 degrees so that the square symmetry is easier to see. Now, can this be verified? We have just predicted that if I take a platinum crystal and I cut it along 1, 0, 0, I should get a square surface. Can I check that the atoms are indeed arranged according to a square symmetry? And here I will introduce one of the tools of surface science. And this is to show you how you can have some atomic level information on a real surface. And this tool is scanning tunneling microscopy. Now, scanning tunneling microscopy is based on several technical developments. The first one is the use of piezoelectricity and piezoelectric devices. What you have here, this would be your sample, your flat surface sample, and here you have a very sharp tip that is mounted on a piezoelectric tripod. If you control the potential difference along one of the arms, you can make it shorter or longer. In other words, you are able to displace the tip in all three directions of space, parallel or perpendicular to the surface. And that's one first technical development, because some people had to realize that this was precise enough to control the position of the tip with a precision of a fraction of an angstrom, smaller than the size of an atom. Another technical development, which doesn't seem like something very high-tech, but which is quite necessary. If you want to have atomic level resolution, you need a very, very sharp tip. And this is a microscopic view of a cantilever used in STM with the tip here at the scale of the micrometer here and at the scale of the nanometer here. So the tip is really here, and you see that its radius is really only a few atomic diameters. So they had to develop some electrochemical means of sharpening the tip in order to have a very precise tool for microscopy. And the last component of the STM technique is the so-called tunneling effect, which is a characteristic of matter at the quantum physical level. This is how it works. Suppose you have an electron here in the tip described by a wave function, and suppose that you have a very small gap, maybe a couple of angstrom, between the tip and the surface of the sample. Well, in classical physics, the electron would stay in the tip because there is no contact. In quantum physics, things are a little fuzzier, and actually, the wave function will decrease exponentially, but if the surface is really very close, it will not fall to zero. So the electron might find itself in the sample. It might have crossed the vacuum gap, even though its energy was not enough to overcome the energy difference of the barrier. So that's a quantum effect, and it's very strongly dependent on the separation D between the two components. The probability of the electron getting through is exponentially dependent on minus, you see, minus 2D, and then you have other factors involving the energy gap and involving some constants like the mass of the electron and Planck's constant. But this is the important part. If you have many electrons, you will have an electric current. The probability of a given electron going through the gap will be given by this. And if you have many electrons, you will have this factor times the number of electrons going through. So you can measure a current between the tip and the sample that will tell you how high the surface is, how close to the tip. And since, as I told you, you can control the tip positions in the 3 dimensions, you can scan the surface in the y direction, in the x direction, and for each point, you measure the tunneling current, and you will get a map of the surface. It will be actually a set of numbers, intensity of the current for each position, but you can translate it into a kind of photographic image using a color code. Let's say that if you have the atoms close to the tip, meaning if you have a small d, you will represent this in white, and if they are further away, you will represent it in black. And in this way, because you control the position at more precise level than an angstrom, you will get an atomic level map of the surface. And now if you do it on the platinum surface, platinum 100, as you remember, you expect it to be square, and this is what you see. It's already a quite old result, but it's quite spectacular because this is not square at all, right? This is a single platinum atom, and around it you see a set of 6 other atoms disposed on the corners of a hexagon. This is a 6-fold and not a 4-fold symmetry. And so you see that things are happening. What happened here is a surface reconstruction. Why this happens is another matter, but you can see that there is a high level of complexity that you can observe on this surface. In addition, I would say that this is interesting too. You have a regular arrangement of platinum atoms, all the little white spheres, and from time to time there is one missing. This is a vacancy. This is a defect in the structure of the crystal. And you can see these defects individually because you really have a photograph. It was the first time that people could be able to really see atoms. It happened in the 1980s. Okay, now, I don't know, yes, I'm supposed to stop. We can make a break here, and then I will start again. Yes. Very fine.
Tenemos un break de media hora, por favor. Regresamos a las 12 del día. We have a break. Return to, yes, a las 12.